Hello everybody, I am Deepika from LearnoHub, a free learning platform where you can study physics, chemistry, maths, biology absolutely for free only at learnohub.com. So students, in the last video we discussed about the four phenomena that play a very important role in absorption and conduction of water. Now is the time to discuss the last phenomena, that is turgidity, flaccidity, and we'll also talk about root pressure. So let us start with our topics today. Now talking about our first topic, that is turgidity. So we know that a cell or you can say a root hair, right? So it is always surrounded with some fluid. So in case of a root hair, there it is surrounded by soil which has water in it, right? So as long as the cell sap in this root hair, there is a cell sap, right? As long as that cell sap is more concentrated, so the water will continue to enter inside the cell, correct? Now there will be a stage when this particular root hair cells, they will not be able to accommodate more water. So that state when the cell, no more water enters into the cell, and this state is called as, now the cell is called as turgid, and this condition is called as turgidity, right? So this is turgidity when a cell has enough water and it cannot accommodate more water. So that state is called as, the cell is said to be in a turgid state, and that condition is called as turgidity, right? Now comes the turgor pressure. Now what happens, this is a cell, a plant cell. So now it has a cell wall, it has a plasma membrane, right? So this cell wall will not allow cell to burst when there is a lot of water inside it, right? So it will not burst, but it will actually press the content inside it. Now how will that happen, that we'll discuss. So this is a vacuolar sap, this is the content, this is the osmotic pressure, this is the wall content, and the cell content presses the wall, right? Now what happens when there is a lot of water, what will happen, this content will now need a lot of space, so it will start putting the pressure on these walls, right? So now, but the cell wall is very rigid. It does not allow the the inner content to move away from the boundaries, right? So now what will happen? This cell wall will put pressure on this cellular content, and it will press the content into the cell, right? Into the center of the cell. It will keep on pressing. So there are two pressures that are acting over here. One is this cell content is putting pressure on the wall, and secondly this cell wall is pushing pressure back on this content and keeping this content confined into the cell, correct? So this is called as Turgor pressure, the pressure of the cell content on the cell wall. So that is called as Turgor pressure. So I said there are two kinds of pressure. One is the Turgor pressure, the pressure which this cell content is putting on the cell wall, right? So this content is putting the pressure, that is called as Turgor pressure. Now what do we call the pressure which this cell wall is putting? So that is called as a wall pressure, right? So this wall pressure, the pressure exerted by the cell wall on the cell content and pushing the cell content into the cell, right, into the center of the cell. So this is the wall pressure. So this is how the turgidity of the cell is maintained. Now also a state might come when there is a lot of pressure exerted by the inner content of the cell content. So what will happen at that time? It is very simple. The walls will burst and the content will come out of the cell, right? And there is one more state, that is a balanced state, a state when there is no further movement of the water into the cell. So it will happen when no more water will enter into the cell, and even though the cell sap concentration is far more higher as compared to the outside of the cell, even at that time water, no more absorption will take place, and that state will be called as a balanced state. So now coming on to our next topic, that is plasmolysis and flaccidity. So these terms are very much closely related, but they have a difference in them. So let us consider a plant leaves which are actually aquatic plants, right? The freshwater plants. So when this plant leaf is kept in a fresh water, so that time what will happen? So it is in a fresh water, so the water from outside will enter into the cell, right? Enter into the cells of the leaf. So we will see something like this. So cell in water where the normal turgid condition is seen. So can you find out, can you easily separate out cell wall and cell membrane in this microscopic diagram? So no, right? So this is a microscopic view of that particular leaf, so we cannot uh, distinguish between, just by looking at it we cannot find plasmamembrane and cell membrane individually, right? So what has happened, water has entered from outside to inside the cell, and now the cell is full of water. It is in a turgid condition, it is in a turgid state. So what is the state of the cell over here? That is the turgid state. Now the plasma membrane and cell membrane are lying, plasma membrane and the cell wall are lying close to each other, and now when they are lying close to each other, cell membrane is actually pressing against the cell wall, right? Because there is a lot of pressure from the content that is inside the cell. So in the other situation, in B situation, what we will do, now we will take out this leaf from the fresh water, and now we'll keep it in a solution which has some salt concentration, say 15% of the salt solution or 5% of the salt solution, for a few minutes. Now what will happen? Now outside medium, the medium in which the cell is placed, so say is here there is say 5% salt solution, right? So we have placed this leaf over here. So now what will happen? So since the outside concentration of solute is very much greater as compared to inside the cells, so water from the leaves, the cells of the leaves, will move to the surrounding medium. So what will happen? The cell membrane, now the water has reduced inside the cell, only there is the cell content. Now we will see something like this. Cell wall can be seen now, and the plasma membrane can be seen. Both of them can be seen very clearly, and this is the inner content of the cell. So the protoplasm has shrunken, right? The situation when the protoplasm has shrunken and the cell content moves away from the cell wall, so that is called as plasmolysis. And this state, the state in which the cell is seen after plasmolysis, is called as a flaccid state, right? So this is called as a flaccid state. So I hope this is clear to everyone. So now, if the plasmolysed cell is soon returned to the ordinary water, the fresh water, so it will again gain that turgid state, and this condition will be the deplasmolysed condition, right? So the condition will be turgid, but the process will be deplasmolysis, where again the water from the surroundings will move into the cell, and the cell will again gain that turgid state. Right? So I hope this is clear. So let us now discuss these two terms with these diagrams again. So that will make these terms more clear to you. So first of all, this is a C picture. So this is a fully turgid cell where the water has entered into the cell. It is kept in a fresh water. Water has entered from outside to inside the cell, and this is the vacuole, this is the nucleus, and this is a fully turgid cell where the cell membrane and cell wall cannot be seen separately. They are seen as a one layer, right? So we cannot, if looking at the microscopic view also, we cannot see the cell membrane and the cell wall separately, right? So this is the turgid cell. Now we have taken the cell out of the fresh water, we have placed it in a salt solution. So we can now see that since outside concentration of solute is very high or high as compared to inside the cell, so water will move from inside to outside the cell, correct? So in that case, now since the water is moving outside out from the cell, so the cell membrane, it starts shrinking. You can see it over here. Now we can see the cell membrane at some of the places, right? So in this case, the cell membrane is shrinking, the cell content is moving away from the cell wall. So over here the cell content was pressing against the cell wall, but in this case it is moving away from the cell wall. So it is a condition D. So after sometimes we will see this condition where more of the water has moved out of the cell, and now we can see the plasma membrane at I mean more clearly. And now finally this condition will reach where all the water from the cell will move out of the cell, and we'll get this. So this is the cell wall, this is the plasma membrane, and this is the inner content of the cell, wherein this is the nucleus and these are the vacuoles, right? So this condition, this state is called as a flaccid state. So which state? So this is called as flaccid state or flaccidity, and this cell is called as a plasmolyzed cell, right? So I hope this is clear. So now these three are the important terms that we must understand and we must know because they are very, very important. Now the first one is turgidity. It is a state of the cell when the cell wall is rigid and stretched by an increase in volume of the vacuole. Now if you remember the last picture that we saw, so the cell, there was a cell and the vacuole was taking almost 90% space of the cell, right? So because of the absorption of water. Now the next is plasmolysis. Now it is opposite of it, when the cytoplasm is contracting from the cell wall due to the withdrawal of water when the cell is placed in a hypertonic solution or the salt solution. Now the next term is flaccidity, when the cell content is shrunken and the cell is no more turgid, but the cell is flaccid, right? So these are the important terms that you must know. Now why are we studying turgidity? What are the uses of turgidity to plants? So this topic turgidity is very, very important. So this turgidity, the turgid state provides rigidity to the soft tissues such as leaves. So we know that during daytime when it is very hot, so at that time the amount of water that is being absorbed by roots is very less as compared to the water that is being lost by transpiration, right? So in those cases, usually these leaves, they suffer wilting. So what will happen? They will droop down, right? So there will be wilting of the root. They will droop down because there will be less water. So they will not be that rigid. But as soon as the evening comes, so the transpiration, the rate of transpiration will lower down and there will be more absorption of water, right? So as compared to the transpiration, the water absorption by the roots will be more. So in that case, the leaves will again will be filled with water and they will again be rigid. They will not droop down, right? So this is the first point where this turgidity provides rigidity to the soft tissues, correct? Now the second point, it helps to push through hard ground. So what happens in case of a mushroom, or you can say in case of a seedling? So as the picture is shown over here, so in case of the seedling, what will happen? The seed will try to grow. So there will be some growth from the seed, and this will be fully turgid, right? So it will have a lot of water inside it, and because of which it is going to be very rigid. And this has been seen if you observe around your houses, or sometimes near a concrete floor, or somewhere where there is a lot of these plant growth. So you will see that there are the cracks which have been produced because of the seedlings, and now from that concrete space a plant has started coming out, right? So it helps to push through the hard ground. So can you see how rigid, how this turgidity makes them so rigid that they can even make these concrete walls, and they develop that crack from inside that wall, and they are able to grow under those walls, right, under those concrete surfaces, correct? The third one is turgor. In the root cells, it builds up a root pressure. So we can actually do a small experiment for this. Take a well-watered potted plant. Now cut the stem little above the roots. Now what will happen? So you'll see the water coming out of that stem, right? So you'll see the water is continuously coming out of it. Why? Because the roots are absorbing water. So because of this absorption, this we can see the water that was supposed to move up to the stem, but now since we have cut it in between, so now it is coming out of that stem. So now what happens? In labs we actually do it with a manometer, where as soon as we cut the stem, we will place a small glass tube, we'll fix it with a glass tube, and we'll use a rubber stopper so that it does not come out, the liquid does not come out of it. Now we'll see the rise of the cell sap inside this tube. So what will happen? The cell sap will start rising inside the cell tube, and as the cell sap will rise, we'll see the mercury going up and up. So this will show the roots are absorbing more and more water, and the cell sap is rising. Sorry, the mercury levels are rising, the roots are absorbing water, and this loss of water from the cut stem is called as bleeding, right? So what is it called as? It is called as bleeding, right? So remember this term. The loss of the cell sap is called as bleeding, right? So this is a small experiment that can be done, where this is a well-watered plant, and this was the stem, and now as soon as we cut the stem, we fix it with a glass tube, and we seal it with a rubber stopper, right? So now the water or the cell sap will not come out, but it will actually rise in the tube, and as soon as the cell sap will start rising over here, the mercury levels will also start rising, and this will show that yes, the roots are absorbing water, and there is some root pressure, that is actually this ascent of sap. Basically this rise of the cell sap is allowing these roots to absorb more and more water from the soil. So next point is turgor in the opening and closing of stomata. So talking about opening and closing of stomata, so we know that stomata, they are the tiny pores, right? And which are guarded by guard cells. So these guard cells basically help in the closing and opening of stomata. And now actually the turgidity is also related to these guard cells. So what happens? These guard cells, they have thick walls on the inner sides and thin walls on the outer sides. Now during the daytime, when there is a lot of light, so these leaves, they have chlorophyll, which performs which helps them to perform photosynthesis, right, which is very essential for photosynthesis. So this chlorophyll will absorb light, so carbon dioxide will also come into the leaves, and water is also required, which has been absorbed by the roots. So what will happen, these guard cells will absorb water from the surrounding cells, the epidermal cells, the subsidiary cells. The water is, when the water is absorbed by these guard cells, they become this arc shaped, right? So both the guard cells will become arc shaped, and due to which there is this presence of the pore, there is this opening of the pore which happens because of the arc shaped cells, right? So arc shaped cells basically uh, make the pore to open when the cells become arc shaped. So this leads to the opening of the pore. Now during the night time, so this happens during the daytime, right? So the cells become bean shaped or the arc shaped, and which leads to the opening of the stomatal pore. And during the night, what happens, at the night time, the cells, they lose water, they become from turgid, they become flaccid, they lose all the water. In this case, the guard cells were turgid. In this case, the guard cells are now flaccid, they have lost all the water which they have absorbed from the surroundings. So now comes the next point, that is turgor movement. So the movement in plants is also related to the turgidity. So do you remember a plant you must have seen in your surroundings, that is Mimosa pudica, right? So we also call it as a touch me not plant, right? So it is touch me not. Touch me not, right? So this is a plant. If you touch it, it actually folds its leaves. See, if you touch a leaf very lightly, if you touch one of the leaves very lightly, so in another 2 or 3 seconds you will see that entire leaf, all the leaflets of that particular leaf will fold, right? But if you strongly touch that particular leaf, so you will see that this this effect will spread throughout the leaves, and all the leaves will fold because they will lose the turgor pressure. So at this point of the time, they are turgid, they have water in their cells and they are turgid. But as soon as you touch it, it loses the turgidity and it will fold, the leaflets will fold, right? So this is the effect of touch to the touch me not plant, and this is happening only because it is losing the turgidity. Now the leaves are becoming flaccid, they are not turgid anymore. So what happens when you touch it strongly, this turgidity, this loss of turgidity happens at the base of the petiole, that is pulvinus, right? So when this pulvinus will lose this turgor pressure, this turgidity, so all the leaflets will close, right? So that is why it is called as a touch me not plant. As soon as you touch it, it loses its turgidity and all the leaflets, they actually close. So do you remember that insectivorous plants? So those insectivorous plants, they also close because of the loss of the turgidity. When they are open, they are turgid, and as soon as the insect enters into those leaves, they lose their turgidity and they close. So that is also the effect of turgidity, the turgor movements. So I hope this effects, these uses of turgidity to plants are very much clear to you. So now come the next topic, that is imbibition and turgor pressure together, they generates much force. So first of all, what is imbibition? So it is basically the passive absorption of water by some substances like cellulose or starch. When they passively absorb water, so that is called as imbibition, passive absorption. So you remember, when there is no need of energy requirement to actually put something into the box, right? So into something basically, right? So then turgor pressure, we all know. So turgor pressure is due to the hydrostatic pressure, it is due to the pressure of the water on the cell wall as a result of endoosmosis. When lot of water enters into, see, if this is a seed, it is kept in water, and when lot of water will enter into it, so it will be as a result of endoosmosis. So what will happen, so the cell wall will have a lot of pressure on it because of the water that is inside the seed. So it is the hydrostatic pressure, right? So it will actually, when the imbibition and turgor pressure, both of them are together, it actually leads to cracks, to the bursts of something, right? So you must have observed, if you actually keep a soaked seeds in a box, keep some soaked seeds in a box, so what will happen? So that box will actually burst open because of the imbibition and the turgor pressure that is inside the seeds that are present in the box. Correct? So now the next topic is root pressure. So suppose you take a plant, right, and you cut it over here. So here there are roots and you cut this plant over here. So what will happen? You will see the water coming out from this part, right? So from here the water will start coming out. So how does this happen? It happens because of the root pressure. So basically this is a diagram where we are showing the root here, the cells of the roots. So this is a root here from where the water is entering into the cell. So water will enter from cell to cell and it will move from one cell to another by the process of osmosis. Correct? So from one cell to the other, to the other, to the next, and this way the water will enter into the roots. Now when in the root, so after this it will actually reach the vessels which actually transport water to all the parts of the plant. So what are those vessels called? So those are called as xylem vessels. So finally root hairs will actually absorb water. So through osmosis it will move into the another cell. This cell will become turgid. It will press the next cell over here. So then it will push water into this cell. Then it will become again turgid. It will press this cell. The water will move into the another cell. So this way the water will move from one cell to another cell by the process of osmosis, and finally it reaches these cells that are called as xylem cells or xylem vessels, which transport this water to all the parts of the plant. Correct? Now this is not this is very simple if you talk about a small plant, right? But when we talk about the higher plants, this phenomena is not just that simple. So we will discuss in detail about the higher plants also. So now coming on to our next topic, that is guttation. So what is this guttation? So you must have observed in early mornings on grasses. So grass is a very common thing that is around us. So on grass you must have seen those tiny water droplets on the margins, right? Or on the leaves of certain plants like a tomato plant. So those water droplets are due to guttation. So basically these are the leaf margins and there are tiny pores which are called as hydatodes, and from these hydatodes there is a loss of water in the form of water droplets, and that is called as guttation. So loss of water as water droplets from tiny pores present on the leaf margins. So this is called as guttation, right? And why this guttation happens? So basically when there is a lot of water, when there is root pressure, so due to this root pressure, the water comes out from the stems, from the leaves, leaf margins or the leaf veins. So this guttation is a result of root pressure, right? And from where does this water comes out? From stem, from leaf margins, from hydatodes. Those are the small pores like the stomata, which are the small pores on the surface of the leaves, right? So hydatodes are the small pores on the margins of the leaves. Now, the next topic is importance of root hair and upward movement of absorbed water and minerals. So first we will talk about absorption and upward movement of water. So how does this happen? We know that roots contain root hair. So as you see in this picture, this is the root, and where these are the extensions, these are the root hair, and Where are these root hairs present? In the soil where there is water in the soil. So you can say the moisture in the soil or the water in the soil. So these root hairs have cells which have cell sap, and this cell sap has more concentration of solute, right? So this is the best situation. Like outside the root hair there is a lot of water, and inside the root hair is a lot of solute. So water will enter into the cells, right? So water from here enters into the root hair, and finally from one cell to another it goes on, right? So one cell will become turgid as we discussed, and then it will push the next cell, then the water will move into the next cell, it will push the next cell, then water will move into the next cell. So this is how the water moves, and so finally these water molecules, they reaches the xylem vessel, and from xylem vessel this water is conducted into the stem, and from stem it is sent to all the parts of the plant. So we just now spoke about how the upward movement of water takes place, right? So it takes place by the process of osmosis. Correct? Now talking about minerals. So minerals are usually absorbed as ions from the soil, and this absorption of minerals in form of ions is an active transport. So what is an active transport? A type of a transport which requires ATP, which requires energy. Correct? So there is a dilute solution of water and minerals, and if it is not a dilute solution, and if there is no absorption of water and minerals, so there will be no photosynthesis, right? So since plants need to manufacture food, so for that manufacture plants need to absorb water, and that water has to reach to the top of the plants. This is how plants will get water, and then only plants will be able to manufacture food, right? So where is this food manufactured? In the leaves. And where are the leaves? On the roots? No. If we talk about the higher plants, if we talk about the tall plants, so there is a thick stem, quite a few meters long, and on that there are certain branches, and on those branches there are leaves. So this water has to reach through xylem vessels to all the parts of the plant, especially to the leaves, so that they can actually manufacture food, right? So now we need to understand more about, do roots or plants, do they actually absorb water? Do they actually conduct water? So for that, we can perform simple experiments which can tell us whether they are actually performing that function or not. So our first experiment will be to show whether roots absorb water. So this will be our first experiment. For that experiment, we'll take two test tubes. We'll mark one as A and second as B. So A will be a control setup and B will be actually our experimental setup. So in both the test tubes, we'll fill both the test tube with equal amount of water. We'll mark that level, and after that we will put few drops of oil on the top. Right? So in A test tube, that is our control setup, we will just put this water and the oil, that's it, and we will leave it as such. In B, when after we put water, we put oil, then we will place a plant. We'll pick out one plant from the soil which has its shoots as well as roots intact, right? So we will put this plant into the test tube, and we will leave these two test tubes undisturbed for 1 or 2 days, and then we will observe that the level of water in A will be the same. It is not going to change, it is not going to increase, it is not going to decrease, right? But in case of B, in B test tube, the water level will come down, right? And why we have used this oil, why we have put a few drops of oil? So we have put it to prevent evaporation. So we know that evaporation happens, it is a natural phenomena where if you place one glass of water, if you leave it undisturbed for a day or so, you will see after some time that water level will go down. So basically what happened to that water, the water got evaporated, right? So in form of the water went into the air in form of water vapors, right? So just to prevent that and to actually check the level of water, is it the same or has it been changed, we need to put certain few drops of oil so that water doesn't evaporate, right? So this experiment shows that yes, plants do need water and roots, they absorb water, right? So this was our experimental setup final stage, right? So I hope this first experiment to show whether roots absorb water or not is clear to everyone. Let's see our next experiment. So now talking about our second experiment where we need to show that water is conducted upward through the xylem, right? So we need to understand the vascular bundles. So what are these vascular bundles? So vascular bundles, right? So vascular bundles are nothing other than xylem and phloem, and we know that this is what we have studied, that xylem conducts water and minerals and food is conducted by phloem. So this is what we understood, this is what we learned, right? So let us now experiment the same. So now we need to experiment out that whether it is true that plants xylem conducts water, right? So this is a very simple experiment that we will do. We'll take a beaker, we will put some water in it, and then we will put one dye, that is eosin solution. Basically it will turn the color to this reddish color, reddish brown color. So now what we will do, we will take out a plant from the soil which has its roots intact. Now we will place this medium sized plant into the beaker. It should have its roots, it should have its shoots, it should have its leaves, right? Stem and leaves also. So now we will put the roots completely immersed in the solution, and after 3 or 4 hours, we'll take this plant out. We will take the sections of leaves, we'll make the slide to study the leaves, we'll make a slide from the section of a stem, we'll make a slide from the section of the roots. So we'll make these 3 different slides, and what we will observe? We will observe that there was a portion in all of them which was this reddish color. Why? Because since we know that what we have studied is that xylem conducts water. So xylem was present in roots also. So from roots, it took the water to the leaves of the plant, right? And this is a continuous channel. This is not a broken channel. So basically these kinds of tubes which forms a continuous channel from roots till the topmost part of the plant, so that each and every part of the plant receives water, right? So when we will study these sections under the microscope, we will find these pink stained portions in root, stems and leaves, which shows that there these are basically the xylem vessels which conducted water, and since it was stained with pink, so that is why for some time it has gained that pink color. So that pink appearance is there in the xylem vessels, right? So a very simple experiment, correct? So let us see our third experiment now. So now coming to our third experiment, where again we will see the conduction of water through xylem. So we know that we have studied that if this is, say this is the stem, right? So inner part will have xylem tissues and peripheral parts will have phloem tissues, right? So this is what we have studied, and in depth we will study in higher classes how that happens, right? So what we have to do is, in this experiment, it is a quite delicate experiment where we will take 2 beakers which are filled with water, then we will take 2 plants which has their shoots, but we don't actually need roots in this case. So now we will put these stems inside the water and cut them inside the water so that no air enters into the, when we cut the stem, no air should enter the tubes, basically these plants, okay, the cut parts of the plants. So both of them we will cut them underwater, then we will put a stand, we'll place them, and what we will do it now we will remove the, if this is a plant, right, if this is a stem, say, so we will remove around 3 centimeters of the of this plant, the outer parts of the plant, but the inner will remain as such. As you can see over here, we have removed it in the form of the ring. This is a ringing experiment which is called as girdling, right? So we have removed the outer part of this stem, leaving the inner part intact. In the second case, what we have done, we have removed the inner part of the stem and left the outer part intact. Correct? So we will leave this for another 2-3 days, and now we will observe that the plant where we remove the outer part, so in that plant these leaves were intact, they were fine, they were turgid, and they were standing just like a normal plant, right? But in second case, we will see the leaves drooping. So basically what happened? So basically these leaves did not get water. Why? Because water is conducted by xylem, and we know that xylem is present on the inner side and phloem is present on the outer side. Since we made the cut on the inner side of the stem, leaving the outer parts intact, so the water could not go beyond this point, and water did not reach here, over here, or after. So after this point there was no water, right? So these leaves got drooped. Correct? So I hope this is clear to everybody. So now again we know that yes, xylem conducts water, and xylem tissues, if we talk about a stem, so inner side of the stem are the xylem and outer side are the phloems, right? So another important experiment, so experiment on absorption and conduction of water by plants, where we are now going to experiment to show that food from leaves is conducted in the downward direction through phloem of the stem or in the stem. So if we need to understand about phloem, whether it is present or not, so what we will do, we will take a potted plant, right, and now we will cut the stem in the outer, in the periphery, right? So in this way, right? So inner part of the stem will remain intact, but the outer one we will cut around 3 centimeters in the in form of a ring, right? So now what we will observe, so the observations are very interesting. So leave this plant for as such for another few days, right? So we will experiment that you will see something oozing out from the top, from here. So there will be a cell sap that will be oozing out, that will be coming out basically. So this means that food is conducted from top to the bottom, right? So this we will see as soon as we cut make this ring. We make a cut on the stem and we make this ring. We'll see that there is something sap that is coming out of the stem, right? So this shows that the food is conducted by phloem, because on the periphery is the phloem tissue, and it is conducted in the downward direction because we see it coming out from here, from the bottom part, right, from the upper part basically. So this is the upper part of the cut. And now the second thing that we will observe after some time is that we see this part will start growing. The part of the stem where we made this cut, right, so part above this cut will keep on growing as this part is getting enough food, enough starch to grow. But after some time this part will not grow. The part beneath the cut will not grow, because now its starch, its food will exhaust, so it will not further grow. But the part above the cut will grow as it is getting enough food and everything, right? But how it is possible that plant is able to make food? So plant will be able to make food. There is no hindrance in the absorption and the conduction of water, right? So inner part is intact, right? So it means xylem tissues are intact, so roots will absorb water, and this water will be conducted to the topmost part of the plant, and this way photosynthesis is happening. But beyond this point, the starch is not moving downwards, right? So that is why water is moving upward but starch is not coming down. So this means that a plant, this part of the plant is not getting enough starch or food to sustain itself, right? So this is how we can show that, first of all, phloem is conducting food, and secondly, it is conducting it in the downward direction. Correct? Now coming on to the next topic, that is forces contributing to the ascent of sap. So what is this ascent of sap? Basically the upward movement of water. So what are those forces which actually help the upward movement of water from roots to stem to the different parts of the body, right? So there are basically the four forces. First is the root pressure, second is capillarity, third is transpirational pull, sorry, not full but pull, and last is adhesion, right? So the first is root pressure. Root pressure is very helpful in case of herbs. So basically in case of herbs or herbaceous plants, where the plants are very small, they are lean, they are thin, so in those plants root pressure alone is sufficient to bring the water to the top, right, from roots to all the parts of the plant. Because in this case root pressure basically builds that column which absorbs the water, and it creates that force which pulls the sap in the upward direction, and hence water moves up. Correct? Next one is capillarity. So in case of capillarity, what happens? There is transpiration, right? So transpiration occurs from stomata in the leaves, right? So when transpiration occurs, when the water is lost from the stomata in the leaves, so in that case water is pulled upward in the upward direction from roots to the top of the plant. So in that case a vacuum is created in the xylem vessels. So if these are the xylem vessels, so due to this loss of water, due to this transpiration, vacuum is created in the xylem vessels, and this forces water from the roots to reach to the top of the plant. Correct? Simple. And this force is called as capillary force, right? So what is it called as? This is the capillary force. So also remember, less the diameter of the xylem vessels, more will be this capillarity, more will be this capillary force. So more water will be, more force will be, water will be pulled from lower levels to the higher levels with a greater force. So more the diameter, less will be this capillarity, right? So inversely proportional, less the diameter of these xylem vessels, more will be the capillarity. More the diameter of the xylem vessels, less will be the capillarity or the capillary force. Correct? So the third force that is actually pulling water from roots to the leaves to stem and to all the parts of the plant is transpirational pull. So again, as we discussed, water is lost from the stomata of the leaves to the different to the surroundings, right, to the surroundings of the plant. So in that case, now there is a force called as cohesion force. Which force? Cohesion force, right? So cohesion force is the attraction of a water molecule with another water molecule. So there is a tendency of a water molecule to remain bind to another water molecule, and this is how water molecules are pulled in the xylem vessels, right? So there is a continuous column because all the water molecules, they are attached to each other, and this is how they move in a column, they move in a xylem vessel. So when this transpiration occurs, so it causes more water to be absorbed from the roots, and since there is a cohesion force between the water molecules, so this causes a continuous column from roots till the leaves of the plant, right? And the last one is adhesion. So as we were talking about root pressure, so in case of herbs or herbaceous plants which are very small, which are thin, which are lean, so in those plants, root pressure alone can create that pull, that strong pull which provides water to all the parts of the plant. But in case of tall plants like pine, so it is a very tall plant. So in that case, these plants need to have a continuous column, right? So in those cases, there is one more force, like cohesion force, there is adhesion force. So cohesion force, it was between the water molecules, so one water molecule bind to the another, and this is how they form the continuous channel. But these water molecules also need a force or attraction with the cells of the xylem vessels, right? Then only they will be able to move. If the cells of the xylem vessel and water will repel each other, how will they move in the water column? So it is basically the attraction of water to water molecules to the surface of cells, right, and the water molecules. So it is the attractive force between the water molecules and the surface of the cells. So this is how a continuous column is being created from the roots to the topmost level of the plant, and this is very, very important in case of tall plants like pine. Correct. So these are the four different factors, or different forces which actually help water to move from the lower parts of the plant to the high part of the plant. Correct. So I hope this is fine and this is clear to each one of you. Now next comes is the downward movement, right? So we were talking about how this ascent of sap, this upward movement of water molecules take place, right? But now we also need to talk about the downward movement. So what moves down? Or we can say, or in all the directions. So that is the food. So food, how does that move? So it is relatively simple. If we talk about it, it is just this food or the sucrose, we can say, it get it gets mixed with water. Basically it gets diluted, and this is how simply it flows in the downward direction. So it is relatively simple. So again, a small thing that I can tell you over here is, as we know that if this is the stem, right? So inner side there will be xylem and outer side there will be phloem, right? So water will move out from the xylem into the phloem. It mixes that food or that sucrose or starch, and hence it dilutes it, and it helps in the easy flow of that particular starch or sucrose, right? So it is very, very simple. Correct. So I hope this is clear to everyone. And now just think about a coconut tree. So it is so tall, so big. So basically what happens in case of a coconut tree, when a single molecule of water is lost by transpiration, by the process of transpiration. So in that case, when this single molecule of water is lost, immediately from roots another water molecule is being absorbed by the roots, so as to compensate that one molecule of water that is lost. And how does that happen? This happens by the cohesion forces that are present between the water molecules. So I hope this chapter is clear. This is easy, and now we are going to talk about the questions from this particular chapter. So now the question number 1, we need to study the diagram given below and answer the questions that follows. So this is a diagram where there is a test tube, and we have placed water in the test tube, and on the top there is oil, and then a plant is placed which has roots as well as shoots, leaves, everything. So now the question says that name the process that is being studied in the experiment. So what process are we studying in this experiment? So we are studying absorption of water. by roots. So this is the process that we are studying. So the second question says, why is oil placed over water? So we know that if we take a jug full of water and if we place it on the side and keep it undisturbed for say 24 hours. So after 24 hours, you will observe that the water level has reduced, because it is the surface phenomena. It is a natural phenomena in which water is lost. It is naturally from the surface of the water, right? From surface of any water bodies, correct? So this is what we have studied in our water cycle. So now, why we have placed oil? So reason for placing oil on the top of the water is that it does not allow water to evaporate. So this helps us to understand if the water is being absorbed by the roots or not, right? So uh, it leads to no evaporation. So basically no evaporation takes place from the surface of the water, and hence we can easily study the level of water in the test tube. Now the third one is, why what do we observe in the experimental setup if it is placed in the bright sunlight? So if we place this experimental setup in a bright sunlight, so it will be a very hot condition where plant needs to throw water or remove water by the process of transpiration and absorb more and more water from the setup, or you can say the soil, where this evaporation, this transpiration leads to the cooling effect of the plant. So in this case, the water level is going to decrease. Now the second is humid condition. So what will happen in the humid condition? In humid conditions, the water in the surroundings is a lot, right? So there is a lot of water vapors in the air. So in this case, the level of water will remains almost the same, right? So there is little or no evaporation. Now the third one is a windy day. So what happens on a windy day? On a windy day, there is a lot of water that a lot of wind that is going on, which leads to lot of evaporation, or you can say the lot of transpiration from the leaves of the plant. So in this case also, the water level in the test tube will go down, right? So I hope this is clear. So now the question number 2. So question number 2 says the diagram represents a layer of epidermal cells showing a fully grown root hair. Study the diagram and answer the questions that follows. So this is the diagram where this is the root hair, these are the epidermal cells, and this is the root hair, the extension of the epidermal cell. So there are some labeled parts A, B, C and D. So if you see A, name the parts. So the first part says name the parts labeled A, B, C and D. So we will mark them here. A is the cell wall. It is representing this outer wall, so it is the cell wall. Now talking about B, so this one, so this is what? This is plasma membrane, or you can say cell membrane, right? Now the C is what? This is this is basically epidermal cell, right? So what is it? It is the epidermal cell. Now comes the D. So what is this round part over here? This is nucleus, correct? Clear? Now the B part says the root hair cell is in a turgid state. Name the process that causes this state. Now this root hair is in a turgid state, means it is full of water, it is very rigid. So which process causes this turgid state? So the process over here is osmosis. So what does this do? It is the movement of water from the region of its higher concentration to the region of its lower concentration through a semi-permeable membrane. Correct? Then C part is mention one distinct difference between parts labeled A and B. So what is A? A is a cell wall and B is a plasma membrane, or you can say the cell membrane. So what is the distinct difference between them? So this cell wall is freely permeable, right? And plasma membrane is semi-permeable membrane. So this is the difference between the two. Okay, now the question number 3. So question number 3 says the diagram below represents an experimental setup to demonstrate a certain process, study the same and answer the questions that follows. So this is our experimental setup where we can see there is a thistle funnel, which is inverted and it is covered with a parchment paper. Then there is a sugar solution. There is water inside this beaker, right? So what process are we studying? Which process are we studying? So we are studying over here the process of osmosis, right? So what will happen in this process? So what will you observe in the experimental setup? If this is the experimental setup, so after an hour or so, what will you observe? So in this process, we will observe that after an hour or so, see if this was the initial level in the thistle funnel. So after an hour, and this was in the beaker. So after an hour, the level of the sugar solution in the thistle funnel will rise. So the final level is going to be somewhere here, and there will be slight drop in the level of water in the beaker. Right? Why did that happen? Because over here the concentration of water was more, and this parchment paper allowed water molecules to move through it, but not the bigger sugar molecules to move through it. So what happened? The water from the beaker moved inside into the this thistle funnel, and hence the level of water or the sugar solution, it rose in the thistle funnel, and the level of water in the beaker, it lowered, it became less, right? So this is what happened in case of this experiment. Now, why the sugar solution did not come, flow through this parchment paper? This parchment paper did not allow the molecules that are bigger in size to pass through them. So that is why there was no movement of sugar molecules. So I hope this was clear. Now the C part is what control experiment can be set up for comparison. So what control setup can be set for this particular setup? So for this particular setup, the control experiment is going to be the same, just that in place of sugar solution in the thistle funnel, what we are going to place? We are going to place water only, right? So there will be water in the beaker, and the same water is going to be there in the thistle funnel. Correct? So simple. So next is D part. Keeping in mind the root hair, cells and its surroundings, name the parts that correspond to concentrated solution. So which part of that root hair, cell, surroundings, which is going to be the concentrated solution? So it is going to be the cell sap, right? So it is going to be that concentrated solution where there is a lot of solute. Now, parchment paper. So parchment paper is basically acting as a semi-permeable membrane. It is allowing water to go inside, but it is not allowing the sugar molecules to come outside. So this is going to be the cell membrane, which is a semi-permeable membrane. Now, the third one is water in the beaker. So what will be the water in the beaker? It is going to be the water in soil. Right? Now, next part is E part. Name any other material that can be used instead of the parchment paper for the above experiment. So, the above experiment was what? It was osmosis. So, for osmosis, we always need a semi-permeable membrane. So, what another semi-permeable membrane that can be used? So, it can be a cellophane sheet, right? Cellophane paper, or it can be animal bladder also. So this is the question number 3, and I hope it is clear to each one of you. Now the question number 4. So question number 4 says a leaf cell of a water was placed in a liquid medium other than pond water. After some time, it assumed the shape as given below, right? So what has happened over here? This is the cell wall, this is the plasma membrane, and this is the inner content of the cell, and we can see the content has shrunken, right? So what has happened? Plasmolysis has happened, right? So what is the term for the state of the cell that it has acquired? So which state is this? So there are two states, either it is a turgid or it is flaccid. Turgid is when it accumulates lot of water from the surroundings and it becomes very rigid. But when cell loses water, so it becomes what? It becomes flaccid, and the plasma membrane and cell wall, they move away from each other. So basically plasma cell membrane get moves away from the cell wall. Correct. Now the next is name the structure which acts as a selective permeable membrane. So which structure over here act as a semi-permeable membrane? So it is the plasma membrane. Correct. So which membrane is this? Plasma membrane. Now the next comes over here is C, the comment on the nature of the liquid surrounding the cell. So what kind of the liquid is basically surrounding the cell? So what causes the cell to shrink or give away the water that is present inside the cell? So that means that solution is hypertonic. Which solution is this? The hypertonic solution, which means the solute concentration of the solution in which the cell is present is more as compared to the content inside the cell, right? Name any feature of this plant cell which is not present in animal cells. So very simple. In animal cell, there is no cell wall, right? So there is no cell wall. Simple. Now, E part, redraw the diagram of the cell if it is soon placed in the ordinary water after some time. So what will happen? So say if this was the cell wall, right? Now, the cell membrane will come close to the cell wall. The vacuole inside it is going to become big as it will accumulate more and more water. So there will be nucleus and there will be lot of content, the cellular content inside the cell. So it is now going to be fully turgid cell. Now talking about question number 5, which says give reasons root hair become flaccid and droop when excess fertilizers are added to the moist soil around them. So what happens when, if you see, if this is the root hair, so these are the epidermal cells, right? And from here there is an extension of a root hair. Now these root hair, they become flaccid. I mean, which means actually when the cells of the root hair, they lose water and they become drooped, right? So why does that happen? When we add fertilizers to the soil, so the water, the solute content of that water increases. So which means the solution becomes the hypertonic solution. Now there is more water outside, more water inside the cell than outside the root hair, right? So water will move out of the cells, and now the situation will come when these cells of the root hair, they become drooped, right? So they actually wilt, kind of a wilt. If we talk about in case of leaves, so they actually wilt, right? So soil water becomes hypertonic due to addition of excessive fertilizers as compared to the cell sap of the root hair. Now what happens over here? The plasmolysis take place. The root hair becomes flaccid and drooped, right? Simple. The solution becomes hypertonic, and the water from the inside the root hair, the cells inside the root hair will move outside the cell. Now they become flaccid. They lose their turgidity. Correct? So I hope this is clear. Now comes the question number 6. Diagram given alongside is the diagram of an experiment at the start to prove a particular phenomena. Study the same and answer the questions that follow. So the question number 1 is name the phenomena being studied. Which phenomena is being studied? So if you first study the diagram, so this is a U-tube which is being separated by a semi-permeable membrane. This is tube A, this is tube B. In tube A, there is 10% of the sucrose solution. So basically these are the sucrose molecules which are in more number, and this is tube B which has water molecules and it has 5% of the sucrose solution, right? And it has more of the water molecules. So if you study this, since it is separated by a semi-permeable membrane, so which phenomena are we studying? So it is always wherever there is a semi-permeable membrane. So this means what we are doing, we are talking about osmosis, correct? Second is the solution in limb A hypertonic or hypotonic? So in limb A, there is limb A, there is limb B. So is the solution hypertonic or is it hypotonic, right? If we talk about limb A, so in this particular solution, there are more number of solutes. So this is going to be hypertonic solution, right? As compared to B, this is a hypertonic one. Then 3 is which limb of in YouTube is functionally comparable to the root hair in the plant. So root hair, so when we talk about root hair, we say that it has more concentration of solute. So in that case, which tube has more concentration of solute and which tube is going to absorb more water? So that will be tube A. So water will flow from B tube to A tube, right? So this is more concentrated one, right? So it is the answer of this part is tube A, correct? So I hope all the questions that we have discussed today, all the topics that we have covered today are clear to you. So now we will meet in our next video wherein we'll start with the next chapter. Until now, take care, and as you all know, LearnoHub free है पर best है.